Hello, this is an educational video on Tidal CO2. We'll be discussing what Tidal CO2 is, how it is measured, and the importance it has in various medical applications. Tidal CO2, or EtCO2, refers to the concentration of CO2 present at the end of a person's exhaled breath. It is typically measured using a device called a capnograph, which uses a capnometry sensor. The sensor is responsible for detecting the concentration of CO2 in the exhaled breath, and the specific number that can be observed on the monitor is taken from phase 3 in this 4-stage waveform. The waveform can be used to assess a patient's respiratory status and identify issues such as ventilation abnormalities and airway obstructions. The capnograph's waveform illustrates precisely how much CO2 is present at each phase of the respiratory cycle. Keep in mind that the waveform's amplitude or height represents the concentration of EtCO2, and the width of the waveform represents the inspiratory, or breathing in, and expiratory, breathing out time. With that in mind, let's take a look at this wavelength. The wavelength consists of 4 different phases. Phase 1. This occurs just before inhalation. Notice that the EtCO2 level is at 0, and no CO2 has been exhaled. Phase 2. The CO2 rises from 0 and approaches its maximum concentration as it passes the sensor. Phase 3. The concentration of CO2 is relatively constant, and note that the end of this phase marks the end tidal CO2 measurement. So in this phase, phase 3, this is where we actually get our end tidal CO2 number that shows up on the monitor. The last phase, phase 4, this is where the start of inhalation begins. The CO2 waveform returns to 0 as there is no CO2 being exhaled at this point. Remember, while assessing the 4 stages of the waveform, the CO2 concentration should always start at 0 and always return to 0 with each breath. If you're not observing this, you must assess and determine why, because something may not be right. Capnography assesses a patient's real-time ventilation, with ventilation referring to the amount of air that enters and leaves the alveoli. With every breath of atmospheric air, or the combination of gases, which include oxygen, a person takes in, their lungs will exchange gases between the inhaled breath and those that are being channeled through the lungs. Now, let's quickly circle back to this idea of capnography being real-time ventilation. Capnography is known as real-time ventilation because with every single breath exhaled by the patient, a measurement of CO2 is being observed and can be used to consider the patient's current status. While ventilation refers to the amount of air that reaches the alveoli, perfusion refers to the amount of blood flow to the alveoli. Adequate perfusion is necessary for proper gas exchange to occur in the lungs. This is a process by which oxygen is taken in and CO2 is eliminated. Abnormalities in perfusion, such as those that may occur during CPR, can actually be detected through changes in EtCO2 levels. So you can actually look back on EtCO2 readings and see when the patient initially coded, when the CPR began, when the provider may have become fatigued and needed someone else to jump in, all the way until when ROSC first appears. Pulse oximetry, SpO2. Pulse oximetry can be used to assess perfusion and how oxygenated the patient is. It uses an infrared light to shine on RBCs, red blood cells, to determine their size. Generally, an RBC's size can vary depending on if its heme group is occupied by oxygen. A well-oxygenated RBC will signal the RBC to contract or shrink in size, whereas a RBC that does not have O2 will expand or enlarge until its heme groups are occupied once again. It's important to note here that an RBC will either contract or expand despite what is actually occupying those heme groups. So it's an important consideration because carbon monoxide will occupy a red blood cell's heme group and prevent oxygen from attaching. Carbon monoxide poisoning is a serious danger to people as the carbon monoxide molecule has a significantly greater attraction to a red blood cell than an oxygen molecule. Thus, even comparatively small amounts of carbon monoxide can lead to many issues as well as death. Sedation. Sedation scales which are used to assess the levels of sedation in a person often incorporate EtCO2 measurements as well. The Richmond Agitation Sedation Scale can be used to assess a patient and subsequently rate them based on varying levels of either sedation or agitation. A patient who is alert and calm is rated as a 0, as they are neither agitated nor sedated. Moving up this scaling system, you can observe increasing levels of the patient appearing uncomfortable. The RASS has a max upper score of positive 4, and this represents a patient being combative and a danger to others. Moving down the scale from the baseline of alert and calm, you can see that a patient's response to stimuli decreases as the score drops further. A patient with a maximum lower score would be completely unarousable and have no response to voice or physical stimulation. A possible example of this is a patient who is being deeply sedated for a medical procedure. Patients receiving any type of sedation will benefit from having their ETCO2 observed. With an Alaris pump, you can add the ETCO2 module to monitor a patient's ETCO2 levels. In order to do this, first, apply the bi-flow nasal cannula to the patient. Next, attach CO2 to the Alaris module. This is the yellow end. Next, attach the O2 line to the oxygen on the wall as needed. This is the green line. Lastly, from the Alaris pump, select the ETCO2 and switch to monitor. The PCU will now display the ETCO2 and the patient's respiratory rate. There are many factors that can either elevate or decrease ETCO2 concentrations in the body. Broadly speaking, an individual's metabolism can do both of these things. If a person is experiencing pain, it is expected that hormones such as adrenaline will be released in response. This can increase the individual's metabolism and lead to an increase in carbon dioxide production, which can quickly be observed in the ETCO2 reading. Shivering can also cause elevated ETCO2 levels. This is because the act of shivering requires an increase in metabolism because the body is trying to generate heat to warm itself, and that process requires energy. Conversely, and in regards to metabolism, hypothermia can be a cause of decreased ETCO2 in a patient. This is because at low temperatures, the body will slow many processes down, causing a decreased demand for the metabolism to be at work. Metabolic acidosis can also contribute to lower ETCO2 levels in the body. This is because typically, metabolic acidosis is caused by an imbalance in the body's acid-base balance. This then leads to a decrease in the pH of the blood, making it more acidic. The body tries to counter this often by increasing the respiratory rate. This is an attempt to blow off more CO2 and decrease the HCO3 levels, which will try to then neutralize the acidity. Colorimetric capnometers. A colorimetric capnometer consists of a pH-sensitive impregnated paper that attaches at the end of an endotracheal tube. The color of the pH paper changes from a purple to yellow when the capnometer detects carbon dioxide. Colorimetric capnometers can be helpful in assessing whether or not an endotracheal tube placement has been successful. Also, when a patient is undergoing CPR, a colorimetric capnometer can be the first sign that the patient is attaining ROSC.